Hello everyone, today I am going to talk about homeostasis, which is a chapter 14 of CIE, Biology. Let us begin with the definition of homeostasis. Homeostasis is the control of an organism's internal conditions, such that it's always kept close to a constant. You should memorize this definition because many past papers had been asked to write a definition of homeostasis. When surrounding temperature decreases, hypothalamus releases a hormone that stimulates the anterior pituitary gland. The anterior pituitary gland secretes the thyroid-stimulating hormone. The thyroid-stimulating hormone stimulates the thyroid gland to release thyroxine into the blood. Thyroxine increases the rate of metabolism, which increases heat production in the liver. At the end, when surrounding temperature increases back to normal, the hypothalamus reduces the release of thyroid-stimulating hormone so that less thyroxine is produced from the thyroid gland. Next, deamination is the removal of amine group from the amino acid in the liver. Amino group forms ammonia, and rest of the amino acid forms acid. Ammonia combines with carbon dioxide to form less soluble toxic substance urea. This is a structure of kidney. You should be aware of this structure as you already studied about this structure in IGCSE. If you forgot, please pause the video for a moment and draw this picture on your notebook for memorizing. This is a structure of nephron. Like a kidney structure, you should also be aware of this picture. Now, let's talk about ultrafiltration. In the glomerulus, afferent arteriole has bigger lumen than efferent arteriole. This creates a pressure inside glomerulus. Big molecules such as proteins cannot be filtered by basement membrane. Some other solvents like glucose, vitamin and amino acid are passed into nephrons, but soon reabsorbed through selective reabsorption in the proximal tubule. These are three main factors that affect the ultrafiltration. Selective reabsorption happens in proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. This process happens with sodium-potassium pump first. Sodium-potassium pump in the cell lining the proximal convoluted tubule moves sodium ions out of the cells into the blood. Sodium will diffuse out of the fluid in the tubule into the cells using co-transporter proteins in the membrane. As sodium is transported, the co-transporter also transports another molecule such as glucose or amino acid. Glucose moves from the cell back into the blood. Why the concentration of urea in the fluid in the nephron increases as it passes along the proximal convoluted tubule. Water is reabsorbed because substances such as amino acid and glucose are reabsorbed into the blood. This increases the water potential in the fluid in the tubule and decreases the water potential in the blood. Therefore, water moves down the water potential gradient by osmosis. Osmoregulation is the control of the water content of the fluid in the body. First, hypothalamus detects the change in water potential. Hypothalamus is the site of ADH production. When water potential is low, posterior pituitary secretes ADH into the blood. ADH combines with the receptor in cell surface membrane of collecting duct. This activates an enzyme and vesicles containing aquaporins. This increases permeability of collecting duct and water reabsorption increases. Insulin is produced when blood glucose concentration is high. Insulin is produced by beta cells which are made in pancreas. Insulin combines with the receptors of cell surface membrane of muscle cells. This activates an enzyme and vesicles containing GLUT move forward and fuse with the cell surface membrane. Glucose molecules then diffuse through GLUT by facilitated diffusion, and this decreases the blood glucose concentration. Glucose is then used for respiration and converted to glycogen. These are the tests that can be used for diabetes. To explain more about the dipstick's role, dipsticks have enzymes called glucose oxidase and peroxidase. Glucose oxidase catalyzes the oxidation of glucose. Hydrogen peroxide is produced. Then the peroxide catalyzes the reaction between hydrogen peroxide and colorless chemical compound, forming brown compound. Therefore, brown color indicates that there's glucose in the blood and person is suffering from diabetes. The darker the compound, more glucose present in blood.